Hi, I'm Jaebum Park, who is a graduate school student in the Plant Breeding and Genetics Department of the Cornell University. Today, I'm going to present one of the efficient methods for QTL analysis of the tetraploid potatoes with the title, New Users' Experience with the Tetraploid Map and SolCAP SNPs for QTL Analysis in Potato. So before I start my presentation, I wanted to introduce my experience very briefly. As explained in the development of the SNP-based tetraploid maps for potato webinar, the tetraploid map is very powerful tool for QTL analysis. However, about 2 months ago, I hesitated to use and learn this program since learning and using new program seems to be long and tedious work. At the same time, the number of the SolCAP SNPs, 8,303, made me feel overwhelmed. So I'm really worried about that, but the truth was totally different. For the most part, the using and learning tetraploid map was easy. So today, I'm not going to cover every details of the tetraploid map. Instead, I will emphasize 3 primary challenges that I faced during the research. Also, I will introduce some interesting research that I found. For this research, I prepared the Y36 population, which comes from the cross between NY121 and NY115. And the total progeny number was 207, and 1-year phenotype data were used for this. And for genotyping, I used the SolCAP 8303 SNPs. And the program that I'm going to focus on today is TetraploidMap, which was designed by Dr. Hackett, Malin, Bradshaw, and Wu. So here are the primary challenges that I faced. First one is converting original data into the appropriate format for TetraploidMap. And second one is moving any marker that have been placed on the wrong chromosome. And third one is phasing, which means deciding which of the 4 homologs each marker resides on. So you do not need to worry about understanding every details at this point, since I will give more explanation for each categories. And this picture shows overall outline for the QTL analysis of the tetraploid potatoes. And the 3 primary challenges that I faced happen in the 3 red boxes: data preparation, ordering, and constructing linkage map. So in the data preparation, I faced the first challenges, changing data format. Right after genotyping with the SolCAP 8303 SNPs, I got this type of the data. And here are details. As you can see in the first line, there are names for each clone, including 2 parents and the progeny. And from the second row, the marker name is recorded, and the genotype codes where A and B are 2 SNP alleles at a locus are recorded next to the marker name. However, the appropriate data set for tetraploid map is like this shape. So as you can see, like the girl, you can see some difference between appropriate data and original data. So let me introduce the details of the appropriate data first. In the first row, as you can see, there are progeny number and loci number. And in the second row, the marker name and the number of the alleles are recorded. And in the third row, the genotype codes for 2 parents and the progenies were recorded. And from the fourth row, the pattern of the second row and third rows are repeated until the last marker. So now you can see some difference in the genotype code shape and the format compared to the original data. So to get an appropriate data set, 3 steps are needed. First one is changing codes from letters to numbers. And second one is reformatting data. And third one is saved as a text file. And I will give more details for each one. First step, changing codes from letters such as ABBB, AABB to the numbers 1 or 0. In this case, I assign 1 and 0 for the presence and absence of the allele A, respectively. So as long as the genotype codes include the allele A in it, I assign 1. And in other case, I assign 0, like the second example. And to change from letters to numbers, I use the replace function in a Microsoft Excel, and I could easily do that. And if you have a missing value, you can put the number 9. And second step is reformatting data. As you can see, except the difference in the genotype code shape, you can see some difference in the format. For example, when you see the original data, the marker name and the genotype codes for the progeny and parents are on the same row. However, when you see the modified data, you can see that the marker name and the genotype codes for progeny and parents are on the 2 different rows. So that means I need to change the format. So if I had a relatively small number, I might change manually. However, I had 8,303 markers. Reformatting 8,303 markers one at a time by hands seems to be a disaster for me, like this girl. So it was a big problem for me at first. So some researchers suggest creating new program by using R package or PER, or maybe you can create a lot of equations by using the Excel. However, to make a new program, I had to spend at least a couple of months to take a computer programming class and to think of algorithm of the new program. So instead of doing that, I spent around 30 minutes to change the 8,300 markers by using macro function in the Microsoft Word. Let me briefly explain the characteristic of the macro in a Microsoft Word. If you use the macro, you can save some orders belonging to the Microsoft Word into your computer memories with the keyboard shortcuts. So to use the macro, you just need to copy and paste in the original data into the Microsoft Word, and then just click the record new macro icon under tool and macro categories. After that, you just need to click keyboard icon. And then you just need to assign the keyboard shortcut that you want. In this case, I assigned Command, Control, and H. And this slide is pretty important. Now you need to change the format with using keyboard only. So first, I add a chromosome number into the marker name. And second, I add a number of the alleles next to the marker name. And third, I put the marker's name and the progeny data on the 2 separated rows. At this point, I wanted to briefly talk about the work adding chromosome number into the marker name. Actually, the adding chromosome number is not necessary for data preparation. However, by doing that, I could do easily constructing linkage map later. So I will give more explanation for the advantages of the adding chromosome number into the marker name. After that, just click the stop recording. So now you store these 3 orders into your computer memories with the keyboard shortcut. So from now on, you just need to push the keyboard shortcut to convert until the last format of the last marker. So here is one example that I did. To save the time, I changed the first 10 markers manually and store the keystrokes in a macro. So after that, I just push the keyboard shortcuts 830 times only to change the 8,300 markers. So I know it seems to be pretty ugly work, but it works. So because I took less than 30 minutes to change 8,303 markers. So this is your choice. You can choose creating new computer program versus using macro. After that, I could get complete format for the Tetraploid map. And please do not forget saving as a text file, since Tetraploid map can recognize the text file mainly. And at this point, I wanted to talk about selecting marker issue very briefly. as explained before, one of the limitations of the Tetraploid map is that only 800 markers can be loaded at once. So when I start this research, it seems that Tetraploid cannot load enough number of the markers for QTL analysis. However, in practice, it was not much of the problem with the SolCAP SNP chip, because around 10% of marker in a cross between the two tetraploid potatoes are informative. In this case, the meaning of the informative markers is simplex or duplex and present in only one parent, like this example. So let me show my example. In my population, 427 markers were informative in parent 1, and 316 markers were informative in parent 2. So when I consider informative marker only, I realized that the number of 800 load marker was not a small number anymore for QTL analysis. Additionally, when I used more than 430 markers, I could not save the result. I mean, I could get a result, but I could not save that. So if you consider these two issues, this limitation is not a big problem anymore. From now on, I'm going to explain from clustering to the constructing linkage map. To help your understanding, I prepare understanding of the issue terminology. I prepared this picture. Right after importing marker data into the Tetraploid map, the markers are mixed to each other like the first picture. And through the clustering, I can collect the markers which are on the same chromosome group, like the second picture. As you can see, there are 12 different chromosome groups, and markers are collected based on each chromosome group. And through the ordering step, you can rearrange the marker which are on the same chromosome in an order. And then through the phasing step, you can decide which of the 4 homologs each marker resides on, like the last picture. So in the clustering, you just need to put the number 12, since potato has a 12 chromosome. And right after clustering, I faced the second challenges. Please look at the left side picture. As you can see, the red markers which came from the chromosome 7, and the yellow markers which originated from the chromosome 2 are on the same chromosome. So this is kind of a problem. Clustering is performed by many calculations of the computer. So depending on the data set, the result can be wrong sometimes. So in this case, I need to move any markers that have been placed on the wrong chromosome. Please look at this example. In this case, the marker comes from the chromosome 2, 7, and 1 are on the same chromosome. So to move these markers, I just push the Ctrl key plus double clicking. I could get this move marker window, and at here I could move the marker that have been placed on the wrong place manually. And at this point, I want to talk about the advantages of the adding chromosome number. If I did not add chromosome number into the marker name, I need to compare this result with the reference of the SolCAP 8303. So that means a lot of comparisons are needed. However, by adding the chromosome number into the marker name, I could scan and find out the markers which are on the wrong place easily. So I think this example well reflects that reflect the advantages of the adding chromosome number into the marker name. After that, just click the order icon for ordering, and at here you can choose the method among 4 options. Generally, if the chromosome group, if one chromosome group includes more than 51 markers, you cannot use the Ripple ordering. And generally, simulated annealing ordering takes too much time compared to the other 3Ds. Right after ordering, I faced the third challenges. Once marker have been allocated to a chromosome, deciding which of the 4 homologs each marker resides on is needed. This step is called as a phasing. And I think this step is relatively difficult for beginner. To do phasing, you need to examine all pairs of the markers by considering SC group and further information box. And if necessary, you can use recombination frequency and LOD score. I will give more details for each one. Generally, SC group shows the simplex markers which are closely linked on a same double helix of the DNA. So please look at this example. If you find the markers which have the same SC group codes like the first 6 markers, you can interpret them as the markers are closely linked on the same double helix of the DNA. So I think this is pretty useful tool for phasing. And second tool is further information box. You can see this box in the middle of the screen. And when I enlarged it, I found some special codes like 1000 next to the marker name. So to use the further information box, you need to understand some concept of the this box. So for example, if you found the codes in the first picture like 1000 and 100, these codes are corresponding to the left side picture. So as you can see, 2 markers on the same chromosome. So I can conclude that the 2 simplex markers are linked in coupling. And if you found the codes in the second picture, like 1100 and 1000, and these are corresponding to the left side picture, and I can conclude that the simplex marker is linked in the coupling with the duplex marker. And this picture shows the recursion phase. For example, if you see the codes in the third picture, like 1000 and 0001, these are corresponding to the left side picture. And I can conclude that simplex markers are linked in recursion. And if you found that the codes in the last picture, you can imagine the left side picture, and we can conclude that the simplex marker is linked in recursion with the duplex marker. So let me show some examples. In this case, the ones for each markers are on the same column. So I can say that the 2 markers are on a coupling phase. And this example also shows the coupling phase. And in this case, the ones for each markers are on 2 different columns. So I can say that the 2 simplex markers are on a recursion phase. And this also shows the recursion phase. And in this case, it shows the recursion phase between duplex marker and the simplex marker. And this also shows the same thing. And in this case, it shows the recursion phase between duplex marker and simplex marker. So if necessary, recombination frequency and LOD score can be used. Generally, they can be used to assume the maximum likelihood estimate of the most likely phase. So if the further information box and SC group could not give enough information for the phasing, you can use these 2 things to exclude uninformative markers. So as you can see, the right side panel shows each actual value for recombination and LOD score for each marker, and the bottom panel shows the graph of the recombination frequency and corresponding LOD score. And after that, by using the phase information obtained by SC group and further information box, you can put the information on chromosome location of each allele at the phase column. So, for example, when I compare the 4 markers which are checked with the colored arrows, I found that they are on a recursion phase, like the right side picture. So as you can see, those markers are recursion phase on the picture. And to get a complete linkage map, you need to illustrate this shape into the phase column. So for that, I assign 100 for the first marker, and 0100 for the second marker, and 0010 for the third marker, and 0001 for the last marker. So after doing that, I could get the complete linkage map. And this picture shows the chromosome 1 linkage map. And this is for 4 and 5. And while observing the complete linkage map, I found some interesting things. As you can see, there are huge gaps between the markers like this. And when I saw the chromosome 6, there are huge gaps between the two markers. And the chromosome 10 also has the gaps. So just before I said there are large gaps between the markers on many homologs. And I think they may limit the ability to detect QTL. So I think further researches are needed to explain these gaps. Nevertheless, when I used the Tetraploid map, I could get some significant QTL peaks. So this will reflect that the Tetraploid map is still a powerful tool for QTL analysis. And now I wanted to briefly talk about the last step, QTL analysis. Compared to the other step, QTL mapping was relatively easy. So if you read the manual, you can easily follow that. And results are following. And this is when I focused on the chipping color, I found some significant peak on chromosome 7 and 2 and 10. And interestingly, the result of the chromosome 2 and 10 were similar to the papers written by Dr. Dauchis and Fryer. And when I focused on specific gravity, I found significant peak at chromosome 4 and 5. And the result of the chromosome 4 and 5 were similar to the paper written by the Bonnier et al. So now I wanted to talk about the other issues that I encountered. First of all, depending on your computer and data set, you may or may not be able to load the data. You may or may not be able to load the Excel data directly. So when I tried to load the Excel data into the Tetraploid map, I could not do that, but some researchers can do. So I cannot guarantee if you work with the Excel file. However, if you work with a text file, it always works, so you do not need to worry about this issue. Also, as explained in the many conference, it can be learned only in Windows XP. So I tried to use in the Windows 7, but I could. But the truth was, I could install the program into Windows 7, but I could not use many functions of the Tetraploid map. So I highly recommend using Windows XP. And let me conclude my presentation now. At first, new users may hesitate to use and learn Tetraploid map because it seems to be pretty difficult and complex if he or she just glanced the manual once. However, based on my experience, it was not difficult and complex. So I wanted to say that once you learn about this program, future QTL analysis for the tetraploid potatoes will be fast and easy. So thank you for listening to my presentation. And I want to say thank you for my major professor, Walter Duong, and my lab member. And also, I appreciate the help of Dr. Dauchis. Thank you.